Next, certain diseases caused by the viruses. First one is mumps. This is caused by paramyxovirus. Smallpox is caused by variola virus. Herpes is caused by herpes virus. AIDS is caused by HIV. Influenza is caused by myxovirus. These are some examples of diseases and the viruses that cause them. Some symptoms of plant diseases. Symptoms in plants include symptoms such as mosaic formation, leaf curling, leaf rolling, stunted growth, dwarfing, leaf yellowing, leaf vein clearing, etc. These are the symptoms of plant diseases. Next, we are going to do the structure of two important viruses, that is TMV and T2 bacteriophage. Tobacco mosaic virus and T2 bacteriophage. So what you're looking at is the tobacco mosaic virus, also short formed as TMV. It's just a section of this long virus. As you can see, this capsid on the outside is a helical capsid, and these individual units you can see repeating all over, and inside this you see coiled inside here, this is the nucleic acid, which will be single-stranded RNA. Tobacco mosaic virus was discovered by DJ Ivanovsky. Is credited with the discovery of the viruses. This is the first virus to be discovered, tobacco mosaic virus, and it's a single-stranded RNA virus. This is how it looks. This diagram from NCERT is important. So this is the structure of the T2 bacteriophage, another important diagram from NCERT. It has a complex shape. As you can see, two types of capsids are found. One is this head. This is the head. The head is polyhedral and it encloses the nucleic acid. Nucleic acid is double-stranded DNA here in this virus. Apart from the head, the rest of the body is made up of helical capsids. Everything that you see over here is made up of helical capsids. This is the collar, this is the sheath. These are called the tail fibers, which are not meant for locomotion. They are just there for attachment to the bacterial host cell. Okay, they are meant for attachment, not for locomotion. Not for locomotion, or maybe they are involved also in all locomotion and we don't know. Anyway, as of now, they are not meant for locomotion. They are meant for attachment. The bacterial cell that it infects is E. coli bacteria. That one only, T2 bacteriophage. So these are the structures of two important viruses, TMV and T2 bacteriophage. We are looking at how a virus infects a host cell and takes over the host cell machinery right now. Look over here, there is a diagram. So this is a virus here, this is a host cell. This is an enveloped virus. As you can see, it is coming and it is coming and integrating with the host cell membrane. It integrates with the host cell membrane such that its envelope becomes integrated with the host cell membrane and the capsid is injected out. This will be the first step, infection of host cell. Once the virus is inside the host cell, see its capsid is intact. What will happen now? Look what is happening. The capsid is disintegrating and the nucleic acid that is there inside is coming out. This is the second step. This is called as unfolding. It unfolds its capsid such that the nucleic acid emerges out. Let's say this is viral RNA. See what it is doing? Viral RNA, this one is single-stranded RNA. What it forms from this RNA is double-stranded DNA. Now the DNA enters inside the nucleus. This is the host cell nucleus. This is an animal cell. It has entered inside the nucleus and the viral DNA is now integrating itself into the host chromosome. Integration into host cell DNA. This will be the third step, viral RNA to double-stranded DNA formation. This is the fourth step, integration into host DNA. Once it integrates itself into the host DNA, it is free to form many RNA copies. It can form many RNA copies. Whose RNA? The viral RNA. For translation purpose, these numerous RNA molecules will emerge out from the nucleus, and once outside into the cytoplasm, they will, using these RNA, they will produce multiple protein molecules. These are viral proteins. These viral proteins are similar to the ones that it already had initially. Now, in the next step, there is going to be an assembly of viral particles. It will assemble all these capsomeres and it will place the nucleic acid inside. There is going to be an assembly and many such viral particles will be produced. Once these viral particles are produced, what is happening? See, this is called budding. In a similar manner how it came and integrated with the host cell, it will pinch out a portion of the host cell membrane and will take it around as an envelope. Now it has become enveloped viruses. This is how the virus will get an envelope around itself, it will bud out and go. Why are these bigger and why are these smaller? Because these are far away. Okay, they have gone. So many of them have come. Fine, this is how the virus will infect a cell. There is an infection of the host cell. There is an unfolding of the nucleic acid. Viral RNA is exposed. From viral RNA, it will produce double-stranded DNA which will go inside a nucleus and integrate itself into the host cell DNA. Once the viral DNA is integrated into the host DNA, it can produce multiple copies of RNA strand, and once these RNA strands are produced, they will emerge into the cytoplasm for translation purposes. The RNA copy is taken and proteins are produced from it. So many protein particles will be produced. Once they are produced, there will be an assembly, packaging of the nucleic acid inside the proteins, assembly of viral particles which will do budding and pinch off from this cell and emerge out of the cell. This is how viruses infect the cell. So this is all from viruses.
महादेव के नाम पे सब्सक्राइब कर दें.